Okay, so hello everyone, and welcome to the first INFOFISH webinar series on technological innovation, focusing today on sustainable aquaculture technologies and exclusively sponsored by Benchmark Group today. It's very nice to see that we're still having participants joining in as we speak right now. And again, we're meeting for the first time this year. So, and this year, INFOFISH will be focusing our webinars on technological innovations. So a year has actually passed by and we meet again, yet still discussing the impacts of COVID-19 pandemic. However, we continue to look for the silver lining amidst these challenging times. And it is undeniable that we are in many ways actually moving even rapidly as we would have even guessed. Technological innovation is in the forefront of driving solutions in the seafood value chain. Mindful of this year, as I mentioned, INFOFISH will be focusing on the series of technological innovations. And it gives me great pleasure to welcome you to the first webinar under this series entitled Sustainable Aquaculture Technologies, sponsored by Benchmark. We have with us today a renowned group of experts who will be presenting to us on the advances in aquaculture technologies. I'm honored to welcome Dr. Audun Lam, Deputy Director, FAO, Fisheries and Aquaculture Division, Rome, Dr. Dorte Laue, Group Marketing Director, Benchmark Holdings, UK, Dr. Martin Rye, Genetics Director, Benchmark Genetics, Dr. Farshad Shiseshan, CEO and founder of Blue Aqua International, Singapore, Dr. Joseph Christopher Reyes, Senior Aquaculturist of the Bureau of Fisheries and Aquatic Resources, the Philippines, and Dr. Barbara Hostins, who is an R&D scientist at Benchmark Advanced Nutrition Business in Bay Technologies. Thank you very much to each one of you for being here today. And most importantly, we are really thankful for your time and to spend with us and the participants here to share your expertise. Without further ado, I will hand over the session to the moderator for today, Mr. Sujit Das, who is the Technical Officer at INFOFISH. Over to you, Sujit.
Thank you, Shilin, for your introductory words. Let me tell you about the objectives of the webinar today. We'll try to address some of the innovative technological solutions like genetic selection, mixotrophic farming in the RAS, and urban aquaponics, and also some other relevant recent developments in aquaculture. Today, we have a distinguished and diverse range of panelists with us. We appreciate your kind presence and request to be with us till to the end and participate actively through the question and answer session. Before moving to the webinar program, I would like to request Dr. Duette Lau, Group Marketing Director, Benchmark PLC, to say a few words regarding Benchmark Group. For your information, Dr. Duette Lau has been working in many global leading multinational companies like Reckitt Benckiser, DSM, and Alltech. And she also holds a PhD degree in analytical chemistry. She is based in Basel, Switzerland. So the floor is yours, Dr. Duarte.
Yep, thank you. If you could stop sharing, then I can share. Yeah, thank you. Yeah, okay. Thanks for the intro and thanks for the opportunity to present Benchmark. Benchmark was founded in 2000 and we are just celebrating our 20 years anniversary. So at the beginning, the founders were concerned with big issues in the animal-derived food production, including antimicrobial resistance, animal health and welfare, environmental impact, and food safety. Very soon, it became pretty clear that aquaculture is one of the most resource-efficient ways of producing animal protein, and it was a young and emerging and promising industry, which has great potential to shape a sustainable future. So now 20 years later, Benchmark operates 3 core aquaculture business areas across the world: genetics, advanced nutrition, and health. And the combination of these 3 business areas allows a more holistic view across the aquaculture industry, simply because it addresses various needs and challenges at different stages of an aquatic animal from different perspectives. Benchmark is a truly global company. We are present in over 20 countries, or put it simply, we have R&D centers, production facilities, and commercial offices in every major aquaculture market and species. Main species we are focusing on are salmon, shrimp, sea bass, sea bream, and tilapia. Our genetics business is represented today in the webinar by Dr. Martin Rye, who will talk during this webinar. Our offer for the aquaculture industry in genetics starts here really at the very beginning of life. We are a leading aquaculture genetics provider who is combining long-established breeding programs for salmon, shrimp, and tilapia with the latest genomic tools and state-of-the-art production facilities. With our genetic services team, we are as active in multiple aquaculture species, which becomes very visible when we are just looking at the world map. Here, the team gained experience from over 30 applied selective breeding programs and over 20 aquatic species across Europe, Asia, Africa, and the Americas. Our footprint in genetics is truly global and constructed for growth. Our land-based biosecure production sites for salmon can be found in Norway, Chile, and Iceland. From there, we supply salmon eggs to customers around the world. And our shrimp genetic facilities are located in Colombia and in Florida. And we are actually becoming and building a shrimp multiplication center in Thailand in partnership with a leading local producer. Our advanced nutrition business, which most of you might know under the name INVE Aquaculture, is represented today in the panel by Dr. Barbara Hostins, who is our biofloc expert. The business was founded over 35 years ago as spin-off from Ghent University in Belgium and acquired in 2015 by Benchmark. With our advanced nutrition business, we are present in Asia, Europe, and Americas with R&D centers, technical support, production facilities, and a sales network. With the portfolio, we are mainly targeting high-performance nutritional solutions and preventative health solutions. Our goal here is to, for example, reduce mortality, improve efficiency, creating better outcomes for producers, and improving sustainability in aquaculture. In terms of product and service portfolio, we include Artemia, so live feed, and Artemia proprietary technologies, specialist diets, and preventative health solutions and protocols such as probiotics, biocides, and water treatments for shrimp and sea bass, sea bream. Focus for us is as a hatchery and also some farm segment. Although not present today in this webinar, our Benchmark Health business is focusing on sea lice treatment and working hard getting innovations into the market. And I hope this gives you all some insights about who is Benchmark, and I'm handing over back to Sujit. Thank you.
Okay, Dankeschön, Dr. Duarte. And it's very interesting that Benchmark is contributing in the aquaculture sector in various ways, including Benchmark genetics and in bee technologies and other sister concerns. So before moving to our next panelist, I'd like to remind all the participants some of the housekeeping rules. You can dear participants, you can participate actively in the webinar by asking questions and sharing your views and comments. And for the better interaction, please mention the panelist name to whom you want to interact. We will take some of the questions after all the presentations. And now we are moving to our next panelist. Today, our first panelist is none other than Dr. Odun Lamb, and he's from FAO headquarters, Rome, Italy. And welcome, Dr. Odun. Just a second. Welcome, Dr. Odun.
Good morning and good afternoon, good evening, depending on where you are. First of all, thank you to INFOFISH for organizing this. I think it's very exciting to be here and see the agenda you have and the focus on innovation and technology, which really I find very encouraging. It's absolutely of key importance for the future of this sector. As I told you earlier, I have to leave you in 2 hours, so questions that I'm not able to answer before I leave you, you can of course contact me through INFOFISH if there is anything of relevance for us in FAO. So my presentation today representing FAO will focus on technology and innovation. But I know that the panelists after me will also focus much more in detail on specific aspects of this. So my view is more an holistic, global view as represented by FAO, not focusing on any particular species or any particular subsector of the aquaculture sector. I also want to thank my colleagues who have helped me prepare this presentation. So the first slide, please. Well, first, I'll give you some few slides on the overview of the world of aquaculture, on nutrition, on livelihoods, on volumes, and especially on trends before I go into various aspects of technology and innovation. Next slide, please. Yeah, I want to highlight 2 very important publications that are FAO publications or publications where we contribute. The first one on your right is the biannual publication SOFIA, or the State of World Fisheries and Aquaculture. And I really recommend it to any of you who is interested not only in aquaculture but also fisheries. It presents the state of our sector and also very important information in terms of statistics on state of stocks or aquaculture development, production volumes, values, nutrition, trade, etc. And the one to the left is a joint publication between FAO and OECD. It says their agricultural outlook, and it's a 10-year perspective. That of course is correct, but more than agriculture for us, it also includes fisheries and aquaculture. And it shows the projections for volumes and values over the next 10 years. So you'll find a number of important trends displayed in this publication. Next, please. Well, this is a very important but famous slide that you've seen many times. It shows, of course, the importance of fisheries and aquaculture, but in particular the growth of aquaculture and how it's aquaculture production and volumes that really is driving the volumes and supply in our sector. We know already that aquaculture now has overtaken that of capture fisheries in terms of contribution to human consumption, so fish food consumption, but also in absolute numbers, the trends show that aquaculture will overtake capture fisheries in absolute numbers in 3-4 years from now. So that is very encouraging, but of course there are also some challenges to that and constraints, and we'll look into that later on. Next, please. Well, this shows the development in aquaculture production. Overall very positive, but it's also a question that we should address here, because we see that the increase is not homogeneous. We see that it's Asia that is driving aquaculture development and growth. We see very promising developments also in other regions such as Africa and especially South America. But overall, both Europe and the Americas, both Europe and the Americas have a stagnation in the aquaculture sector, and in fact a decline when you look at over the last 20 years. And that of course is a challenge to all of us. It's also related to policy, of course, and competition with other sectors, and we'll go into some of those constraints later on in my presentation. Next, please. Well, this shows the same slide in terms of growth in aquaculture production, and it goes by species. What's important to notice is that it's all subsectors are increasing, especially inland aquaculture, which is very, very important, that not always recognized outside the sector. Thank you. Next, please. Consumption, of course, a record 156 million tons now coming from capture fisheries and aquaculture. Again, it's aquaculture growth that is driving the supply. The slide to the right here shows that aquaculture now has overtaken that of capture fisheries, as I mentioned, in terms of not in absolute numbers, but in its contribution to consumption. Because we know capture fisheries also has significant volumes still go to feed production that not all goes to comes back to the sector. Next, please. We also have to remember that the fisheries and aquaculture sector sustain millions and in fact indirectly hundreds of millions of livelihoods of people around the world. So not only in terms of consumption of food, but also its livelihoods, its jobs. And this includes also aquaculture, very important. The role of gender is not always recognized. In fact, we don't really think about gender and the involvement in women maybe when we talk about capture fisheries and fishing, but even in capture fisheries and in the primary sector, including aquaculture, 14% of those who are engaged are women. But if we include processing and trade, 50% of the involved, the labor force in our sector is women. Now that is not recognized at all, and it's something that we really have to think about, and we have to improve the awareness of that, and also, of course, to improve the conditions of all those who are engaged in the sector, whether it's aquaculture or capture fishing. Next, please. So the real topic for this webinar is how to improve aquaculture production and efficiency through uptake of technology and innovation. We have seen that aquaculture has developed very fast recently, in recent decades all over the world, but not similarly or equally in all regions. A couple of regions that in fact are stagnating or even seeing negative growth. There is, however, a huge potential in aquaculture for further growth, much more than in capture fisheries. In capture fisheries, the objective is to bring all stocks up to the maximum sustainable yield, and that could possibly yield another 20 million tons in capture fisheries. But in aquaculture, of course, the potential is very large. It's very large. I'm not going to put any boundaries here to the growth, but I think it's only human imagination and the market that really will set those constraints down the road. So technology and innovation has to be, our efforts have to be problem-oriented and also demand-oriented. We have to focus on what the consumer wants, what are the needs of the consumers, in order to satisfy those needs. In fact, I think the aquaculture sector, at least previously, was too much focused on production rather than producing for the consumer. But that has much improved, and in fact, all through the value chain, whether it's capture fisheries or aquaculture sector, there's much more attention on what the consumer, on what the market really wants and is looking for in terms of solutions to their needs. Next, please. There are a number of limitations, of course, regarding the resources available, land and space, and the land in this sense can also be the water bodies, of course. There's increased competition with other economic sectors, whether it's tourism or construction or agriculture. There is a limited access to water resources in many countries in the world, many areas. In many areas of the world, when we talk about food production, agriculture is much more important and dominates very often policy, and therefore aquaculture not always has the role in development and food production policy that it really deserves, especially when we look for the future. There is also issues related to feed, whether it's fish meal or fish oil, but it could also be other feeds coming from agriculture. For example, in salmon, we see the number of issues of soybean production coming from unsustainable sources. And it's fairly labor intensive. And of course, the fact that it's labor intensive has also resulted in a number of innovative approaches in reducing the need for labor in many areas of the world. But of course, that also depends on the cost of labor in the various countries. But overall, it's fairly labor intensive, and in many areas of the world that is also a constraint, but leading to technological innovations and solutions to remove that constraint. Next, please. Environmental concerns, some of the most important ones, the limitation carrying capacity of the systems, a number of issues related to environmental sustainability. I mentioned fish meal and fish oil. There is the issue of use of chemicals in drugs, not only in the sector, but also in other sectors that have an impact also on the water bodies in which we operate. Climate change has its own challenges, disasters related to changes in weather, and of course the last one, last but not least, COVID. And just one issue, I think it's very important to address all the concerns that are out there also in the mind of the consumer. Very often the image of aquaculture could be much improved. A lot of people have issues with aquaculture that are not related to facts, that are based on perceptions, wrong perceptions, but still they are influencing policymakers, and therefore they are influencing also the choices of investors, of banks, of insurance companies, and the community at large. I think as a sector, we have a huge task to undertake to improve the image of the aquaculture sector by focusing on facts, information, being transparent of the problems we have, but also pointing to the solutions that are out there. Next, please. Well, vulnerability and sustainability, we mentioned that, and there are a number of issues related to this, and there are a number of solutions. We have, for example, better management practices, good agriculture practices. So there are a number of solutions, guidelines, and instruments that are used in the sector to overcome these issues of vulnerability. Next, please. Next slide. Yeah, FAO has been working on the ecosystem aquaculture approach or ecosystem approach to aquaculture for a number of years. Can you go back, please? It's a holistic approach, and in a few words, it attempts to integrate all the issues in a holistic way by being participatory, by focusing a lot on spatial planning, that not seeing the sector in isolation, but planning and operating the aquaculture sector as a sector within a space that is also legitimately used by other stakeholders, whether it's agriculturalists, whether it's tourism, whether construction. The point is that everyone has to find a well-defined role within this system. And it works well in most environments and in most contexts. However, it has its limitation when you have a number of stakeholders and when the reality is very, very complex. But it's a very powerful instrument for planning and implementing successful agricultural operations within a defined space. Next, please. But here I've just mentioned a number of issues. I will not go into them in detail. I mentioned already the issue of land and space, the environment, and a number of sub-issues. The issue of breeding and genetics. My colleagues on the panel will go into that later on. Biosecurity issue is increasingly important, of course, and paid attention to. Labor issues and labor-intensive practices, and therefore innovation through engineering and mechanical solutions, and also the role of business and new business practices new distribution and sales channels, whether it's e-commerce or other types of distribution that they've seen over the last year. We've been driven to increase innovation also in sales and distribution through the COVID pandemic. Next, please. So I mentioned the issue of aquatic feed and feeding and some of the more innovative and recent approaches, the increasing use of plant protein, for example, in the feed formulation. Now also of insects. Insects have also been, but beyond the feed issue, insects have also very recently been approved in Europe as a direct food. So these solutions are becoming more and more interesting and encouraging, also identifying new sources of protein. As live food organisms, Artemia, etc., the improved formulations, which is an ongoing, of course, it's a trend. There's a continuous improvement in feed formulations, of course, and also the integrated multi-trophic aquaculture aspects and solutions that are seeing more and more. Increased precision in feeding, leading to more efficiency, but also less cost and less waste, and also filtration of solids. So there are a number of solutions. These are just a few of the more innovative practices, but there are many, many more that we'll hear about later on. Next, please. In terms of hardware, of course, there are a number of new systems, whether they're indoors, on land, offshore. In salmon, for example, there's huge investments going on right now on land salmon farming facilities that still have to come to market in volumes, but of course it will have a huge impact also on the overall supply in the sector. And if this is successful, it could also be replicated in other in the production of other species. There's also a long-standing practice of integrated aquaculture and agriculture, especially in rice production and certain carp or tilapia species in Asia. And we have also environmentally friendly aquaculture with improved pond systems, standardizations, and more efficiencies. We got the pond raceway aquaculture technology. We got a number of the use of robots, for example, in the monitoring of water, but also, for example, in the cleaning of nets. I mean, they're incredibly important and encouraging uptake of technology over the last few years. And every time you go to an exhibition now, you remember when there were physical exhibitions, there was incredibly exciting offers of new technology in all aspects related to production, not only in the aquaculture sector, but all sectors related to water management and water policies. Next, please. And of course, things are getting, or equipment is getting sturdier. It's getting sturdier, more resilient, more robust for a number of reasons, in order to improve resilience as such, in order to be able to go further out in the water, in the ocean, or further down, and also to be more resilient in terms of the changes of climate. You see in more and more extreme weather situations, storms, floods, etc. So it really is important also to improve the resilience of the equipment that we engage with. And we see the number of improvements also in size recently. And here are some of the examples there. Next, please. Well, species diversification, that is obvious, and that is taking place. But at the same time, I think that also in the future we'll see that most of the species that are farmed, we will not see a huge number of species. We will focus on a fairly limited. I mean, The potential in terms of number of species is of course enormous, but I think because of economies of scale in resource and feed, in production, in the equipment, etc., and not to, and also the mind of consumers, I think we'll see a fairly limited number of species that the sector will focus on. So I don't think that will change very much. We'll see new subspecies, we'll see some diversification overall, but I don't think we'll see a very large number of new species being found. Next, please. I think what we will see is a lot of improvement in the farming of the species that we already have. Integrated agriculture, especially with rice, I mentioned the use of aquaponics is encouraging, and also the integrated multi-trophic agriculture practices. And also in Europe now, aquaponics is getting a lot of attention and fairly encouraging, but limit on a limited scale on the project. It's an interesting process. Next, please. No, next, please. This is too technical, I think, for this seminar. We'll take the next one, please. So, aquapark construction and management, an interesting concept, especially in China. It helps the national planning process by enabling policy and targeting certain areas where you have aquaparks, which include aquaculture, but also a number of other economic areas. So it's very much related to the ecosystem approach in aquaculture, spatial planning, taking into account the available land, water, etc., and well defining who's doing what within this aquapark context. Next, please. Yeah, these are some example of aquaparks in China, and you can see that a number of economic activities are taking place. Aquaculture, yes, but also not only the farming, the rearing, but also there's the hatchery, there's the feed mill, there are the ponds themselves, there's a hospital. So a number of issues or economic areas related indirectly to the production of fish or shellfish or whatever. There's also the component of training, of educational facilities linked to this, and also ecotourism is encouraged. So people are encouraged and come and learn to see, and this is also improving the awareness, of course, but also the acceptance of aquaculture in a larger context. Next, please. So there's a number of synergies of aquaculture with tourism. We've seen that in Asia, but also increasingly in Europe. We've seen similar initiatives with capture fisheries, with small-scale fisheries, but I think it's also something you have to do a lot more with aquaculture. It's an economic subsector in itself related to tourism, but it also improves the awareness and the acceptance of aquaculture as a legitimate economic and food-producing sector of quite some importance in most areas. Next, please. So some of the important aspects of best aquaculture practices in a number of sub-sectors there related to broodstock, seed, cage aquaculture management, fish feed, biosecurity and fish health management, the management of effluents and waste, discards, transportation and storage, marketing, of course, and a way forward towards the both the FAO Code of Conduct on Responsible Fisheries and also the implementation of the SDGs, Sustainable Development Goals, which focuses a lot on, of course, also sustainable food production, in which aquaculture plays a huge role. In fact, there is no way the SDGs can be achieved in 2030 if not the aquaculture sector is continuing to grow and to supply increasing amounts of food to the world population. Next, please. So finally, the way forward to create an enabling environment is fundamental. It's fundamental in terms of having the right legislation that permits aquaculture development, investment, that is attracting private sector engagement in aquaculture, and will bring it forward. We saw already that there are at least two regions in the world where aquaculture production is stagnating and in fact declining. That is Europe and the Americas, North Americas in particular, not South or Central America. And that is something we have to address and turn around if we want those regions to continue to contribute to food production coming from aquaculture with important numbers. There's the governance on sustainable development. FAO is developing guidelines on sustainable aquaculture now that will be addressed at the FAO subcommittee on aquaculture subcommittee next, well, at the end of this year. Continued support to research and development, the fundamental role of the private sector, and of course, in order to attract finance, credit, investment, we need to have a sector that is attractive, that has a correct perception in the minds of investors, of consumers, and everyone. The issue of standards and certification is becoming more and more important. Certification not only for environmental parameters, but also certification related to the well-being and the decent working conditions of those who are engaged in the sector. Disseminate technology worldwide, learning from each other, region by region, South-South, or Asia helping Africa, South America, whatever. We have to learn from each other. And of course, the issue of international regional cooperation and partnership. And I think this webinar was an example. So thanks to INFOFISH again for taking this initiative and in fact helping disseminate knowledge and facts about the sector and also about technology and innovation. Next, please. So just my final slide here in FAO, I'd like to mention three important events. We got the Committee on Fisheries, which meets every 2 years. It's meeting now in a virtual meeting in Rome in 2 weeks' time, with policymakers from all over the world also discussing issues related to aquaculture development. There is the very important global conference on aquaculture that we do every 10 years. It was supposed to be last year, but it has been postponed and will take place in Shanghai in China in September this year. It's every 10 years, so it will focus on the achievements over the last 10 years and trends, and then setting the scene for what has to take place in the sector in order to continue to grow and face those challenges that we have over the next 10 years. A very important goal, especially because the next 10 years up to 2030 are the objectives and the goals, the targets of the SDGs, the Sustainable Development Goals. So we'll also focus on what the aquaculture sector can do in order to achieve the SDGs by 2030. And the subcommittee on aquaculture, which will take place in Mexico at the end of the year. Thank you. Next, please. So with that, I thank you for your attention, hoping I didn't get too much out of the timeline, but thank you again for organizing this information.
Okay, great. Thank you, Dr. Odun, for your very interesting updates. I think aqua park, aqua tourism, and governance and sustainable sustainable development, many things you have discussed, many interesting technologies also. So as you have another webinar, so we would like to ask one quick question before leaving, and that is we see in the market fourth generation aquaculture technologies are evolving, and many new technologies are coming. You already mentioned that robots are coming in aquaculture waste treatment. So what effective strategy do you think will increase the aquaculture adoptions in Asia Pacific region?
Well, I think it's very difficult to give some standardized answers that apply to everyone. I mean, we talk about the aquaculture sector, but of course it's a very diverse sector and a number of species. The different countries are in different development stages of aquaculture development, and also they're very often at different stages in the uptake of technology. What I think is important to remember is that innovation is fundamental. We cannot stand still, and the sector is not standing still. The sector is continuously innovating. The sector is continuously taking on new technology. What I think is important to remember that new technology is not always high tech. It's not always very expensive. There are a number of things that can be done in terms of innovating your practices that is not necessarily very expensive. I mean, you have countries where labor costs are very high, and of course, therefore, you need to invest in labor-saving practices. But there are many countries of the world where that is not the case. So you can innovate without a lot of investment in a number of areas of the world, and new uptake of digitalization of a number of techniques, technologies in Africa, in areas of Asia, which is not necessarily a high investment. So I think that is important to remember, that the sector is incredibly diverse, and there are different solutions depending on the development stage at which you are. I think that is important to remember, but also that innovation as such will trickle down. So innovation in the most advanced areas within the sector will also trickle down and lead to efficient and cost-effective solutions also in other areas of the world. So I think the sharing of information and the fact that innovation will sort of be spread, we'll start somewhere but spread, I think that is very encouraging and will.
Yes, thank you for your answer. So I think, and if we can move to our next panelist. If we got time, we will come back to you. I would like to remind all the participants that you can answer the poll questions. Poll questions are there, and kindly your kind feedback will help us to get the feedback on the webinar. So we request all the participants to respond the poll questions. We are now moving to our next panelist. Our next panelist is Dr. Martin Rye. He's the Director of Benchmark Genetics. Dr. Martin has more than 30 years of experience working in genetic research and applied selection of salmonoids and other aquatic species. And he graduated from the Norwegian University of Life Sciences and holds a PhD degree in animal breeding. And he has working experience with the NOFIMA, and he also played a central role in establishing Aquaphonics genetic centers, which is a part of benchmark genetics right now, and it is since 2015. As the director, Dr. Martin take care of the development of benchmark genetics activities, and he also facilitates the strain development of benchmark for salmon, shrimp, and tilapia. So it's over to you, Dr. Martin.
Thank you, Sujit. Let me just get my presentation up.
Yeah.
I hope you see this one now.
Yes, all right.
Yeah. First of all, I would just like to thank you for being able to give a presentation of this topic of genetics. I titled this one "Applied Genomics: Supercharging Selection for Disease Resistance in Shrimp." And basically what I'm going to talk about is primarily some of the trends that we have seen in the technologies applied for genetic work in aquaculture species. But I do have some very recent and unique examples on how these technologies now on genomics can be applied or are being applied for crustaceans with the Vanamai. I would just like to bridge first to what was said by Dorte about benchmark genetics. She was mentioning that we are working now across 3 major areas for development of sustainable aquaculture productions worldwide. Along with genetics, we also work with nutrition and animal health. But at this point, I would just like to say that genetics is strongly linked to development of sustainable production. Not only by the improvements that we are making on the feed resources and the utilization of the resources that we are putting in, not only feeds, by the way, it's also the land and the water. And it also plays a very critical role for improving animal welfare. By the work that is done on disease resistance, for example, it creates a much better situation for the animals that we are farming. And also, of course, we are contributing significantly to the economics of the aquaculture operations, and that is also a part of the sustainable aspects of doing this type of production in the first place. So what I'm going to talk about now is based on the trends, as I said, is the trends on development of technologies applied to aquaculture species. Benchmark is in quite a unique position because we are running in-house now 5 state-of-the-art breeding programs for aquaculture species. We are running 3 programs for salmonids and have a very strong global footprint, approximately closing in at 30–40% of the total production has a footprint from benchmark on what's coming out of the salmon genetics materials that are distributed. Then we are running own programs in shrimp and tilapia as well. And besides that, we also have very extensive experience from the other species, like what Dorte was pointing out. And by having this type of framework we are working in, we have a unique possibility to deploy technologies that are developed for some of these species onto other species. And in particular in salmon, where a lot of development of genetics is the most advanced programs going on, we take the opportunity to take some of the tools that we have developed for salmon onto these new species like shrimp and tilapia, for example. And we are in a unique position to be able to do that efficiently. Now, let me just outline a little bit of what is the toolbox we do have for generating genetic changes in these target population we work with. The standard now for the industry in aquaculture is this type of design I'm just outlining here. It's a Sib Selection Scheme. And this engine, it consists of a breeding nucleus in which you are keeping your breeding candidates. You are reproducing in every cycle a high number of individual crossings in the hundreds. And then you are taking a subset of all those families you are producing, and you can put them into a range of different performance tests. And typically what we are testing for is growth performance in different production environments. It can be the survival recorded in different environments. We have traits that are linked to carcass quality. And of course, now we see a very rapid uptake and focus now on improving the animal's ability to withstand pathogens. That's becoming a very key aspect of many of the most advanced programs in the world. So this breeding nucleus in the center here is kept under a very high biosecurity, in most cases the nucleus. And then the SIB testing that is on the left side of the screen is happening outside this nucleus. And it's important to say that the engine consists of this centerpiece and the testing. And it generates incremental improvements in these populations that we are targeting. Typically what we see is that we can generate from 5 to 15% improvement per cycle. This depends on which species we are talking about, which type of traits we are working with, but also how many traits we are trying to improve at the same time. And all the changes that are coming in the breeding nucleus, it's permanent changes in the population. So over time, you can produce very significant improvements on these animals that you are putting into this type of system. And now, before talking about the genomic tools that we are developing now, let me just demonstrate a little bit on the power of this fundamental design I just showed you. What I'm going to show you here is data, actual data from a tilapia program that has been running for many, many generations. And what you see on the screen here is the starting genetic variants that we started with in this population. And you can see now when we are going from one cycle to the next, how we are gradually improving the whole population. We are moving the whole distribution on the underlying genetics in a favorable direction. And you can see now two things. Number one is that over time, there are very big differences happening and improvements happening in this population. And secondly, if you do these things in the correct way, you can see that you are maintaining genetic variance as you go. And that is a key point, to find the balance between the short-term selection response and maintaining genetic variability. But if you do this in a sound way, in a scientific way, you can create a system that has the ability to go on for many, 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 many generations, and doing this in incremental improvements per cycle, and end up with very dramatic improvements in the target populations. Now, you look at this design again, there are some challenges we have. Let's just talk about disease traits. You can see that when we are testing all the nucleus families, we are taking a subset of these animals, and we are from each of these families, we're taking a certain number of animals from each family, and we are putting this into a control challenge test against the target pathogens we are working with. This can be viruses or bacteria. And the issue is that we cannot take animals that has been exposed to the pathogen. We cannot take back into the nucleus and use as candidates. So the only information that we are bringing back to the genetic evaluation happening here in the center of this part of the nucleus is the data coming from the SIBs. So all the candidates that we are keeping biosecure, we only have the SIB information. This means that we can rank the families each breeding candidates are belonging to. But we are not able to identify the best animals within those families, because those animals that has been exposed to the pathogens and survived the pathogen in the challenge test, they are not brought back into the nucleus. So the consequence of this is, for these typical SIB tests in which we only have SIB information, we are basically restricted to select only between families, not within family. So we are selecting conventionally between families, which is working on 50% of the genetic variance. And that's why these traits are the core target, these type of traits for genomic approaches. Because these genomic approaches, they gives us the opportunity now to rank animals without exposing them, in this case, when we're talking about disease resistance, to the pathogens themselves. And just a very quick run through the different development stages. The first application of genomics in the selective breeding setting is called the Qtl selection, or a marker-resisted selection, which is based on a major gene. And the thinking here is that if you have a group of phenotypes, that's what you can measure on the animals. You have animals that are dying and animals that are surviving, if we are talking disease-resistant traits now. And the idea is that if you find one strong genetic marker that is closely associated with a causative gene, then you can use this single marker to identify those animals with the most favorable genotype. So that's a simple thinking about these things, but in a simplistic situation, marker-resisted selection can be very effective if you can find these major genes, if you know that the target trait you work with are affected by a limited number of major genes. And of course, there is a very brilliant case that is from the salmon world, when there is, for the infectious pancreas necrosis, which was a very significant disease in the salmon industry in Norway for many years, we see that there is a single Qtl, major Qtl, with a marker that is explaining approximately 90% of the overall genetic variance. And what you see on the right side here is what happened in the industry with the number of outbreaks of IPN in the industry when these selection on the markers were initiated around 2010. You can see this rapid drop, and this is largely due to this selection, the marker-resisted selection for this major QTL for IPN resistance. So everything looks fine, right? We can just find the gene and we can find the marker linked to the gene and we fix all the problems. Well, it's not that simple, because in most cases, what we typically see is that the target traits that we are working with, they are highly polygenic. That means that you do not find these major single genes that are explaining everything. But in the situation is that these traits are influenced by a large number of QTLs or genes, but each of those have a very small effect. So how do we resolve that situation? That brings us to Genomics 2.0, which is called Genomic Selection. And the principle is basically the same here, because what we have on the left-hand side is a group we call a training data set, in which you have your candidates, you have these exposed to the pathogens in this case. And what you are doing, you are identifying these animals that are surviving and dying. And you do this with a high density marker panel. So you are not only looking for 1 or 2 markers that you are investigating, but you are investigating markers in the thousands. And the key point is, if you have a high number of markers spread across the whole genome, and if you have a high quality training data or calibration data, you are able to estimate the marker effects on all these small contributions across the whole genome. So what you are doing then, you are picking up the QTL effects of all these small minor SNPs across the whole genome. And in this way, you can get a very precise estimate on the genetic merit for these animals. And of course, this was brought into the scientific world by a milestone paper that came in 2001. And what we have seen now has been a paradigm shift in selection, in not only in aquaculture, of course, but also in livestock. It happened first in livestock. The first applications of genomic selection happened around 2010, 2011, large scale for dairy cattle. And then we have seen now that the salmon industry is picking up quite quickly. So in 2015, we already had picked up these technologies also in aquaculture. And why are these so important? There are 3 major reasons why this is becoming now the new standard. First of all, I talked about the problem when we only rank families, we can only work on the between family variants, which is 50% of genetic variance. But if you are using genomic information, you are opening up to rank the candidates within the families, even those candidates that have not been exposed to the pathogen, if you are talking disease-resistant traits. So you are growing your basis for selection from 50% of the overall genetic variance to 100% of the variance. And of course, that gives a much stronger selection differential. So you have a better basis for creating genetic changes. Secondly, if you are using the whole information, both between and within family, you can keep up the same selection pressure with the higher number of candidates you're selecting. You don't have to limit your selection to the top-ranked families. You can also find individuals or candidates outside the top-ranked families that are total genetic merit is as high as from the top families. And that's why you can either reduce the rate of inbreeding because you are reducing the relationships among the animals you select, or you can select stronger without increasing the rate of inbreeding. And finally, you can use the true genetic relationship among the animals. Because when we are typically in a conventional selection, we are typically assuming that full SIBs are related 50%, half SIB 25%. But in reality, you have a variance between those groups. And if you're using the genomic information, you can create the true relationship matrix among the animals. And that gives us the opportunity to estimate much more precisely the genetic merit of the candidates. Okay, Benchmark has been a pioneer on taking up these technologies on genomics in salmon. For example, here, we started the very first uptake on genomic selection in 2015 for pancreas disease. That was the first implemented uptake of this technology. And it happened in 2015, only 4 years after this was done first time in livestock. And you can see from this slide that it's rapidly increasing now the number of traits that we are working with genomic information with. Now what we are doing on shrimp, we do have a unique breeding population in Benchmark. We have actually 2 different selection lines. We have one that is resistant or relatively high tolerant to white spot disease, which is one of the major pathogens affecting the global shrimp sector. And we have another one which is more developed for a robust, fast growth line. And when we started now, became a part of Benchmark in 2015, this program, that is the longest standing program in the world in shrimp genetics, started in 1997. That also became a part of Benchmark's operations in 2016. And this gave us a unique opportunity because we were resourced in Benchmark now to take on these new technologies here too. When we started in 2016, 2017, there were no tools available for doing this genomic work. And in reality, what is the vehicle we are using is this Snip arrays, this marker arrays that is the basis for the genomics work we are doing. So what we did in-house was to develop a custom Snip array because there was none commercially available when we started this work in 2017. We started out by setting up, designing an array with 15,000 markers. And we did this in collaboration with Neogen in the United States, one of the genotyping labs. In 2019, we upgraded this to approximately 40,000 markers. And that's the tool that we are currently using for everything that we are doing on genomics in shrimp today. Immediately, we use this high array, this 40K array, to look to see whether we could find some of these major QTLs for white spot. We couldn't do that. We saw that white spot was highly polygenic, which is the situation for most of the diseases and most of the commercially important traits. And that's why we then shifted to look at what can we do with genomic selection. So then we took the opportunity to utilize this huge genetic variability we have in our populations with respect to the susceptibility or tolerance to white spot. So we created an experiment in which we then pooled the whole genetic variance across those 2 populations. We established a training data set, that is a calibration data set for the survival against white spot. And we estimated genomic breeding values, and we looked at the underlying genetic variability for the resistance measured in the challenge test. This was done in a collaboration with Nofima, a research institute in Norway, and partly also supported by the Research Council in Norway. So what we did when we analyzed these genomic data coming from this training data exposed to the white spot, what we saw again is that there was a very significant genetic variability. When we looked at our experimental families, around 70 families that we produced, you can see there is a huge variation in the average survival of these animals or these families. And you see here that the heritability is fairly high. And the same is if you're measuring this not only as the overall survival, but you can also measure this as a resilient traits. How many days do these animals actually survive? In both cases, there is a significant additive genetic variance. And we were using the genomic relationships when we did this analysis. And it showed that the heritabilities that we can obtain by having a more precise relationship matrix is much higher than the heritabilities that we see when we are just using a conventional relationship matrix in this analysis. So then we moved on. And what we did here is that we have now picked up this GWAS, the Genome-Wide Association Study pipeline, in which we can identify major genes We are now routinely using white spot, the 40K for the white spot for EMS. And we also have several other traits in the pipeline now. We already have implemented now genomic selection in 2019 and for EMS in 2020. Now, we built on this experiment that showed these heritabilities. We also did estimate the genetic merit on the genomic genetic merits on the parent generation. And then we created 3 groups of parents that we selected. One with the high genomic breeding values among the parents, one with an average, just a random sample of parents, and the third one with a low one. And what is very interesting here is that we use this to estimate the realized selection response for genomic selection. This is the first time this is done in crustaceans. It was published in Scientific Reports in 2020, and it showed that the realized selection response there is 14%. And that's more than twice as high as any of the estimates for response to selection for white spot that is coming from conventional methodologies. So it's very exciting. It is also showing that we have now similar genetic variants in the offspring generation again, which means that we can continue doing this selection going forward as well. And this is the method that we are now implementing into the product that we are developing now in Benchmark. We are developing growth lines. And of course, we are talking now on the resistant lines using genomic selection. We already implemented for white spot and EMS, and also general pond survival. And then we have hybrids between different product lines. And we are now not using any ablation, eye ablation on any of our broodstock. So that's very interesting too from an animal welfare point of view. And we are also using dry feed, 100% dry feed now on our operations. And of course, that's produced by INVA. And finally, just let me say now, when we have this design, we also have the ability to streamline the product for particular production environments. So on the one side, we do have the BMK Protect material. This is where we are building in the genomic selection for the disease traits. We also have a yield line, which is primarily selected for growth and high survival, general robustness. And then we also have now tailored the material for low salinity. And having this diversity of products is also something that is unique, and it's uniquely brought into the shrimp industry from the Benchmark side. So then I just thank you for your attention, and I'm happy to take any questions in the question and answer session later on. I would just like to acknowledge our core team working with Benchmark Genetics in Colombia. That's where we have our nucleus operation, experimental facilities, and also our NOFIMA colleagues, Dr. Nick Robinson and Maria Lillehammer, who have participated in these genomic studies that we have done, and then my colleagues at the lower left hand here. So thank you for the attention.
So thank you very much, and excellent. It was a very good inclusive presentation. I think all the participants enjoyed and learned a lot from your presentation. For your information, still we see 286 participants from different parts of the world. So I think please stay with us till to the end. We are now moving to the our next panelist. Our next panelist is Dr. Farshad Shishesian, and he's a very known face in our Asia-Pacific aquaculture sector. Dr. Farshad is the president and CEO of Blue Aqua International Group of Companies that has been globally present in around 14 countries. He was also the founder president of Asian Aquaculture Network, and also the former president of Asian World Aquaculture Society Asia Pacific chapter for the 2013-2016 period. He holds a PhD degree in terrestrial and aquatic ecology and has hands-on experience in managing aquaculture farms in different countries around the globe. He also the inventor of a highly commended mixotrophic system, a super intensive shrimp farming system with the PCT patent in many countries. So the floor is yours, Dr. Farshad, and welcome, Dr. Farshad.
Thank you very much. Thank you. Thank you for inviting me. Let me, I just pull up my presentation.
Please.
Okay. You can see my screen. Can you see this?
Yes, please.
Okay. Okay, good afternoon, or I don't know, good morning, ladies and gentlemen, and thank you for INFOFISH and Benchmark for inviting me for this, webinar. I will be discussing today a little bit about, mixotrophic system, but mostly I will focus on the super intensive farming. Just Blue Aqua is a company that has a global presence, and we are in like few countries, and we have a concept. This is important. We have a concept. We call it Dr. Shrimp, and what we are doing here mostly on focusing on the 5 species of the shrimp around the world. And these are like, we are creating a center for the knowledge and all the requirement for people if they want to work on all these 5 species. Also, in terms of fish, we focus on 2 species: is a jade perch and pompano, and also red clawfish and seaweeds. So these are all our focus in a company, so we don't really do other things. Very quick background. I think the previous presenter, they really give a very good picture of the aquaculture industry. But I like to start with a very simple, I usually show this one and talk about this in most of my presentations. Aquaculture industry, it's a very old industry, but maybe old days we don't call it industry. It was just a way of people, surviving and getting the required protein. We have even like development like such a, what you see at the bottom left. It's a largest shrimp farm in the whole planet. It's a 30,000 hectare farm in Indonesia, Lampung. Then we have a lot of nowadays development and a lot of advanced technology come to aquaculture, that even you can see a lot of companies working on AI, automation. So our industry, it's growing very fast from something very, very traditional. But the interesting things about aquaculture is that even today, you can see farms that are very, very advanced, and there are people that they are still working on a very traditional way. As you know, like in our industry, due to the less production coming out of the, less product coming out of the fishery, so there is a demand on the aquaculture. So that's why aquaculture industry is growing. But as other speaker also mentioned, we have a lot of limitation in our in our planet. So intensification and utilizing the correct way of the resources, it's become very, very, very important. That's why we work a lot on the intensification. Okay. Then when you talk about intensification, always people, I'm glad to see like we have colleagues here that they come from a salmon industry. I always believe like the most advanced aquaculture sector in our, globally is the salmon. Salmon people, they have done a great job. And but when we as a farmer, I'm a farmer, when I as a farmer looking at in Asia to the intensive or super intensive, I always picture something like this in my mind. So I always think that super intensive, if I want to do super intensive shrimp farming, I have to follow the footstep of the salmon. Or you can see, like we call it smart farm technology. So we have all these tools, equipment, it looks expensive and complicated. So many farmers in Asia, and we know that Asia is the largest part of the, largest, part of the world that produce the majority of the aquaculture product. So when the farmers look at this, they say, oh my God, this is not what I need. I can't do this. It's so complicated. So same. So you can see there's a lot of tools, equipment, is a engineering, is a biological engineering. When you talk about, this kind of super intensive and farming method. So you need to be because I'm running the similar farm. So we had a lot of challenge at the beginning. So I'm going to share some of our, experience and, the difficulties that we had to adopting the a different system in the species that we are working in Asia. One of the key things that we were, facing, like especially like in Singapore or Indonesia, sometimes when you do greenhouse system, you have a very high temperature, because if you want to do intensification indoor, so you need to close the whole things, and the cost of energy, like I run a farm in Singapore, we have a farm in Singapore, I'll show you later. The things was the temperature under our greenhouse was one of the key things that may cause a lot of problems. So we came up with a very simple, we call it evaporation system. I mean, those people that are familiar with the chicken house, they know this is work in many, many chicken houses. So we try to apply this one in our greenhouse, and there was a great result, out of that. So we solve one of the key things. Then there are other technology. I have here some example, like we call it like air dome or agrodome. These are very good. These are, you can control temperature. You can do a lot of things. The environmental control there. But again, it's the matter of the cost. But when we look at the whole these things, when you talk about the intensification, the whole idea is about very simple fact. We call it carrying capacity. And I always try to summarize this carrying capacity in a very, very simple way, because I give a lot of training and talk to the farmer, local farmer, shrimp farmer, some fish farmer. So when we talk about carrying capacity means what? Carrying capacity means your system, how much you can produce with what you have. So that's always, there are resistance to increase this capacity up. So I simply name it as an environmental resistance. And of course, you have a potential to grow, but how much? I mean, that how much is the key question. So the farmer must understand, if I am doing 1,000 piece shrimp per square meter, it's not because, I do some magic with the shrimp. No, there are a lot of this environmental resistance that must be break down to reach to the capacity. So environmental resistance are a list of the parameters and the factors that always cause a farmer. I always tell a farmer, if you don't have sufficient oxygen, you even don't talk about it. You can have the best genetic, you can have the best water, you can have the best feed, but if your oxygen is not enough, you can't do. So sometimes some of these environmental resistance are very, very key, and we have to look. And it is not that simple. Like, I have a potential, the biological potential, but really how much I can push. I have to really study the water and gas dynamic and understand the correlation between all of them to manage a high density system. So what we came up with is a system, I call it green circulation. So I don't use, and I don't go the way that, for example, the salmon farmer are using. We changed the whole concept. So we came up with a green circulation and a system that contain all the, whatever you can see in a normal farm. So here I just summarize the culture system we have. Of course, in a like very layman word and in like a very, biology 101, I can say you have a water, you have a shrimp or fish, you have a feed, and you have surrounding environment, whatever it is. This is the basic summary. I mean, like there is no rocket science there. That's the whole things we have. So how we can manage this, where what I need and where what I don't need something, like we have to look. So then there is a culture system design. So if I'm doing a super intensive farming, what am I looking for? I mean, like people talk about RAS. RAS, it is not as simple as we think. Like RAS means how many % of water you change. Can you change 10%, 20%, 30%? You can, is it a system that only using like only daily 1% water exchange? Each one of them has own complete different set of the arrangement and protocol. Then one very key things I have seen around the world. This is my farm. We have seen around the world that people use different holding. I call it holding water. I mean, like it is not important, because animal, when you keep the shrimp inside the water, he doesn't know where is he. Is it a round tank or long tank, or is it tall or is it short? So for animal, does not matter that what is the shape of the holding tank, but it's the matter to us that we have to see how we can manage them. So we have a raceways, we have a round tank, short one, we have a tall tank, which is round also. So it's like you have a different selection. How do you choose them? What is the best way of using all these holding, structures? What is the cost? Like, for example, if I open my farm in Singapore, everything is expensive in Singapore. Only air breathing is cheap. Other than that, everything you want to do in Singapore is very costly. So should I use my tank from cement, or I should use from plastic, or I should use I don't know, like this metal structure we have? It all goes back to where you are. What is the material available? How you can do? Then what is the flow of my water? This is the very typical, I think, like you can call it like RAS. Which one I need in my system, which one I don't need. For example, we don't use degassing. I don't use much of mechanical filter. I use a lot of oxygen. Biological filter definitely we use. So it's up to you what you want to exclude or you want to include. I'll show you some pictures that what we have done. Oh, like this, there are some other people, they have a very different approach. I mean, like, using a pump. It's one of the key things that we have in our system here. Because, some of the species we have in Asia, they are not expensive, but you can do intensive farming. So if you want to invest, I don't know, a few $1 million on a farm and you sell something just $2, $3, that's why it would not be possible. So in this farm, which is located in Singapore, is one of our farm, I can produce, like routinely, we can go somewhere around 15 to 20 kilo per square meter of shrimp. I don't talk usually about SR because when you come to the survival rate, I really don't bother too much because I produce my own post larvae. Why I say I don't bother too much? Because to me, my carrying capacity and my goal to have all the way 15 kilo shrimp. If it goes beyond that, I try to harvest. If it goes below that, I leave it. But how many shrimp I put in the tank to get that 15 kilo? It is very, very different question because just now our friend, Dr. Morton talking about the genetic. I think in genetic, after so many years, I have learned some very simple things. One is that growth is really, really related to the environment. Survival, it's related to, the density, the disease, many things. But there is one very, very important factor that when you do high density like us, it's always is a trouble, is the variation of the size. And that is something that somebody like me doing 15 kilo, 20 kilo shrimp per square meter, we face every day. Size variation, it's a very, very important thing because I don't harvest one time and sell one time. No, we harvest every day. So when I harvest every day, I want a certain size of animal. Why I cannot have a uniform size? Because my stocking density is too high in a very short, very small area. Then I have a very different genetic variation among the animals. I mean, like even they come all from the same parents, but still not really same. But it was a monodon time. I used to do farming when monodon was popular. Monodon is different. Monodon, one parent stock, one brooder stock can give you up to 800,000, 1 million post larvae. So you can simply stock in one tank. So they all have the same genetic. But when it's come to the wanami, it's no such a things. You have 100, I don't know, 50, 60, depends on a different brooder stock, and then you collecting no ply, come from a different variation. Even though we think that they are same, but they are not same. And also there is a disease problem here because sometimes, I just give an example, if out of all those brooder stock, one animal is sick, you will not detect by the normal sampling you have. You will have a problem. You may have 1% infection in your population and it's very difficult for you to detect it. And that 1% at the end of the day can give you problems. So what I want to say is when it's come to the super intensive farming in a shrimp, there's a lot of things that it is not only, about the growth or survival. That's what I don't usually care much about the survival. I more care about the biomass. I want to have this biomass with this size. So even if I stock twice, The cost of PL to me, it does not matter because I produce myself, and I don't think so. Cost of PL compared to the whole cost of the operation, it's a very big number. We care about feed a lot. So like this farm, I can produce like every year around 100, 120 ton shrimp from a very small land. The whole land is less than 1 hectare, culture area less than 0.5, 0.6. Okay. We have another farm that is halfway done. So here we have a lot of tall tank. You can see these tanks are around 3.8 meter height. And this is we use for the fish. And we have a raceway. So I use a lot of like we use a robot here, mostly for the monitoring the inside the tank or cleaning. And for the shrimp, very simple. You know, we just use a raceway. We build our own raceway. We have another project in Oman, exactly following the same concept. But the common things about all this project is about simply small land, very high biomass. So let's go back a bit more details on the technical aspect of the things. What are the bottleneck of all these things when we are doing? I think the key things, I want to be very technical here. You talk about oxygen. Oxygen is very important. You can have everything in your system. If your oxygen is not enough, you will fail. Even in an outdoor farm, I have a farm in Indonesia, outdoor, also the same. In indoor, also the same. You go to anywhere, oxygen is the key thing. But people usually ask, how much oxygen I need? A common answer to that, a lot of people say 4 ppm, 5 ppm, wrong. Because when you go to the intensification and you have a high density, it doesn't work like that. It goes by many other factors. What is your BOD? What is your, I don't know, ORP? What is your, I don't know, amount of feed you are giving? How much bacteria you have? What is the water exchange? There are hundreds. And what are the, you know, oxidization rate in your system? That's why it's become more complicated when you have a high density system. I use a liquid oxygen because for such a density, I need to make sure that I have a sufficient oxygen in my system. We use like oxygen cone, or there are some technology from some other company that the whole idea is this: I dissolve the oxygen in the water and I will have a sufficient oxygen. I have an oxygen near to the saturation level. And we adjust the oxygen not by how much we think the system needs. We just follow exactly the system requirement, okay? Then we do a lot of aeration in the system. Aeration is not only for oxygen. I use like aerators that I call it intelligent aerators. So these are very important because aeration in that kind of system is more like a movement of the water. So water must move in any type of system. And other things that we use very commonly is our protein skimmers. Like it's very normal for us. We use it in a closed loop. We don't put in a line, we use in a closed loop because it's very, very important to reduce some of the organic matter in the system. And yeah, we use a robot this is mostly for our fish tanks that they are tall. Some of these robots for monitoring, some of them can use for cleaning because as our friends say, like in Singapore, the cost of manpower is very high. So I can't hire so many people to take care. So some of the things, if we can use the, you know, technology, sometimes when I calculate the cost of one robot like this, I mean, we call it robot, but it is not really robot. They are like a drone underwater, or they have very small capability to really work. Still, you still need a human to manage them. But this will help me in a long run to save money on the manpower. We use an online monitoring system, but again, it goes back to you. What do you want to monitor? Sometimes I always say, too much information, if you don't know how to analyze them, is more dangerous than not having any information. Because there are many farmers, you give them so much information, they don't know what to do. And they get panic and they just make the whole thing collapse. So sometimes information must come with a knowledge. You must know how to use information. If I tell you your alkalinity is low, I mean, I don't care about alkalinity. Why I have to worry about alkalinity? Because people think alkalinity low means shrimp die. It's not true. Alkalinity is nothing to do with the shrimp. Alkalinity, everything to do with the nitrification and buffering capacity and all those. But of course, indirectly impact the shrimp. Then suddenly farmer, they think, oh, I'm gonna fail. But they don't understand alkalinity, many other things, they're all connected. So understanding this information of what information I require, what do I need to take, how important they are, is it has a very long story. You have to go and understand. So we gather information, like these are some of my reading, very simple. I just put a very real numbers here. So we look at like temperature, the DO, ORP, pH, TDS, salinity, but there are other things also we measure. For example, we measure bacterial load, we check for the virus, we check for the BOD, you see like all the things. But then somebody must sit down and conclude what is the meaning of all these things, so that conclusion of the information, I mean, making a conclusion out of information is more important than just having information. My waters all looks like this. In a very highly super intensive system, I have exactly water like this, green and full of foam, but this is what we want. This is all about my mixotrophic system. We do our own post larvae. It's very important. We have even fish, you can see. The density very high, the animal grow very healthy. And this is how we do. In a normal farming system, your traditional farming people can harvest 1 to 2 kilo per square meter. Why I give a square meter? Because I don't have like a pond that is a 1 hectare. So we talk about meter. I mean, our system can do about 15 to 20 kilos. So we basically 1 hectare of our land, it's equal to 10, 15 or 20 hectare, depends on, you know, where you are looking for. That's what I call mixotrophic system. We have a patent of completely, I mean, like, is a granted patent for 7 countries. And we have another system, we call it multi-species super intensive. It's a bit long name, zero waste RAS system. This is what we are doing now. So exactly this is all about circular economy of aquaculture. So we use a waste from one to another one. So that's how we work for so long, you know. And this kind of system work very well in a country like Singapore, or those small country that they have a lot of concern about the, you know, the waste, the land, the water, the energy. So if I want to summarize very quickly here, when we talk about mixotrophic system in a more, you know, science way, there are factors we very concerned, like pH, ORP, nitrogen phosphorus ratio, CN ratio. And this CN ratio, it can be CN ratio for heterotroph and autotroph. They are very important. We work a lot on the bacteria. The application of probiotic is very important. We care about phytoplankton. It's extremely important. It's a totally different from bioflux. It doesn't, you know, really, we don't follow the bioflux steps. A bioflux or flux is a part of my system, but here we really control everything. We work on the nutrient balance, means we look at the nitrogen phosphorus, because by managing a nitrogen phosphorus, I can manage my phytoplankton. I can decide what type of phytoplankton I want. As simple, if I have a 30, means I have green. If I have a 12, I have a brown. If I have a 20, I have a brownish green. When it comes to the carbon nitrogen, is a totally different story. We look at the different level. We add a lot of sugar, or we add a lot of, you know, carbon dioxide in the system. So the nitrogen cycle is extremely important. I have done very layman category here, like phytoplankton I put as an autotroph. Exactly, they are autotroph. Then this nitrogen cycle in the super intensive system that we run is extremely important. I think every farmer must understand exactly how does it work. The other two very important factors is the pH and ORP. We very, very concerned about these two. I'm sure everybody know what is the pH and what is ORP. I'm not going to explain, but ORP is like oxidization reduction potential. It goes by the millivolt, and it's very, very important, even in our green system. Then all this, the different trophic are also extremely important in our system, because each one of them do a different job. So when you look at the ORP is very much related to the, you know, nitrification. So that's why with the low ORP or high ORP, your nitrification will change. And then we look at the phytoplankton management is, again, is an indoor or outdoor, doesn't matter. That's the NP ratio we have there. And then the pH management always come through the nitrogen, come through a phytoplankton management, because phytoplankton is 95% contribute to the pH fluctuation. And the rest is a 5% usually is a respiration. And this is more about alkalinity and hardness. I don't really bother too much about alkalinity, but we look at the, the ions of the minerals are very, very important for us. And probiotic application, exactly, it's extremely important. And this is the stages we have for the mixotrophic. So mostly in an indoor system or outdoor system, we work this way. So in early culture, we have a lot of phytoplankton. I call it phytoplanktonic stage. So in this system, in this stage, we develop a lot of phytoplankton. It doesn't matter is it indoor or outdoor, because animals are still very small, so they fit on this phytoplankton and zooplankton. Then they slowly we reduce the phytoplankton and add more on the bacteria and heterotrophic and partially autotrophic. Then at the end of the cycle, we usually have only mostly our bacteria in the system, because we need to clean the system. We don't bother too much about, but it's still phytoplankton very important, because we need to manage the nitrogen. Okay. I'm going to finish. I think I'm over my time, right? So then this is another method of mixotrophic, which it's more complicated. So I'm not going to explain. This is, we use a nitrogen pick, and then we reduce. So if I want to summarize, like there is a pond management through this. We look at the genetic, definitely very important, the disease, feed management, and environment. Of course, there is a process to do all these things. So again, is it indoor or outdoor? It does not matter. All this process apply, or some of them is more emphasized indoor and some outdoor. I have some pictures here just from an outdoor farm. I show you. You can see like when you kill the phytoplankton or reduce the phytoplankton, you can have a different type of foam. This is not good. This is more because of overfeeding, we call it, but this is good. You have a very nice color of the water, good circulation. You can see it's an excellent circulation of the water, a lot of foam, and the water color here you can see is extremely, full of a bacteria. It's not like a, but it has some, maybe some phytoplankton. We use paddle wheel outdoor as a, as an indication for how this water looks like. And we do a lot of research in Singapore with Tamasak Poly. We have our own R&D department also. I think just nice in terms of timing. Thank you very much.
Okay. Thank you, Dr. Farshad, for the nice presentation. I think our participants learned a lot from this presentation.
Thank you.
So we will move into our next panelist. Before moving to next panelist, I would like to request the rest of the panelists. We appreciate if you could answer some of the questions on the online. So and we will come back to you after all the presentations. If we appreciate if you could answer some of the questions by this time. So we are now moving to our next panelist, Dr. Joseph. Dr. Joseph Sirayos is a fisheries technologist and senior aquaculturist at the Bureau of Fisheries and Aquatic Resources, the Philippines. See, he holds a PhD degree in aquaculture at the Central Luzon State University. Currently, Dr. Joseph is the subject matter expert on aquaponics at the Technical Education and Skills Development Authority, TESDA, and he is also the primary implementer of Urban Aquaponics program under BIFA. Over to you, Dr. Joseph. You are muted. Dr. Joseph, you are muted. Yeah. Okay.
Hello.
Yeah.
Yeah. Can you hear me now?
Yes. Yes.
Okay. Let me share my screen. Yeah. Can you see my screen now?
Yes, please
Okay.
proceed.
So let me proceed. No, the topic that I will be presenting today is the urban aquaponics. Urban aquaponics, an innovative way to ensure food security. So I'm Dr. Joseph Rayos from the Bureau of Fisheries and Aquatic Resources. So what is urban aquaponics? The urban aquaponics program is a priority focus of the Bureau of Fisheries and Aquatic Resources Philippines in support of the Department of Agriculture's Plant program. For the objective is to introduce cost-efficient urban aquaculture technology using a solar-powered aquaphonic system that empowers communities to take control of their food security and give access to clean, healthy food while augmenting income. Next is to accelerate the transfer of technologies in the urban areas to improve productivity of households and small farms, taking into account sustainability and resiliency towards a food-secure Philippines. So what is aquaphonics? So basically, aquaphonics is a combination of aquaculture, wherein you culture fish, and hydroponics, wherein you culture plants in water. So together, that's aquaphonics. So I'll just want to share you some current agri-fishery scenario in the Philippines. So for the past couple of years, there is a declining trend of number of farmers in the Philippines. So the average age of farmers in the Philippines is 50 to 60 years old, and the life expectancy is 70 years old. So hypothetically, if there will be no additional farmers in the Philippines starting today, so for the next 10 to 13 years, there will be no farmers that can plant and produce fish and plants for the people of the Philippines. Another issue is the land conversion. So fish ponds and rice lands are converted into parks, to buildings and malls, and some are converted into highways. So because of this, the area for agriculture and fisheries significantly decreases. So for the concept of aquaponics consists of a combination of aquaculture and hydroponics that grows fish and plants together in one integrated system. So we have fish and the plants in one integrated system. So what's the biological components of aquaponics? Of course, there's the fish, plants, and the microbes. So the fish excretes waste, and the bacteria converts waste of the fish to usable ingredients, nutrients for the plants. And for the plants, it would uptake nutrients from the water, filtering it for the fish. So all components benefits from this one. So aside from the biological components, there's also a physical components for aquaponics. Uh, there are basic parts of aquaponics. First is the fish tank, wherein we culture our fish, and there's the grow bed or hydroplex, wherein we culture our plants. If you could see, fish tanks, we have different design and different materials. Some models uses a concrete, some models uses fiberglass, and some uses tanks, drums, I guess. So, but the concept is there, even though different design, but the concept is the same. So why aquaponics? So number one, it offers opportunity for every Filipino household to produce safe, clean, and accessible food. So that's the most important component why we consider aquaponics. Number two is responsive to climate change. So less gas emission, but same amount of products result. Number three is the additional source of income. So we can sell the surplus. And number four, it is responsive to the new normal way of living because of COVID pandemic. So for the plants of aquaponics, we can use lettuce, tomatoes, pepper, mustard, basil, and other herbs. So we can harvest plants every 30 days for some varieties. So imagine every 30 days you have a new plant, and you can harvest plants. And for the food fish, for the fish for aquaculture, we have two groups. First is the food fish, and then the other is the ornamental fishes. For the food fish, the Bureau of Fisheries recommends the use of tilapia, carp, and catfish. This is due to the fact that all these three species are hardy species, and they are accepted throughout the country. So for the tilapia, carp, and catfish, it is considered food fish and it's target for food consumption. The other group is the ornamental fish, like the gapi, molly, goldfish. For this one, this is intended for selling. So you can see you can use aquaponics for food fish and for ornamental fishes. So advantages of aquaponics. First, reduce water use because aquaponics uses only 85 to 90% less water than traditional farming. Second is can be used on non-arable lands such as desert, degraded soil, or salty and sandy islands. The next one is no need to use artificial fertilizer. Since the plants will consume the waste of the fish as fertilizer, we don't need to add any more fertilizer from artificial fertilizers. Then the plants grow significantly faster. This is due because the nutrients is present in the water 24/7. So the plants can easily access nutrients from the water. Then it shortens the food chain. From the aquaponic unit, you can just harvest it directly to your plates and instant food for the family. But for the traditional way, from the produce from the farm will be transported to the market. That's the only time the consumer can buy. So with aquaponics, it shortens the food chain. Benefits of aquaponics. They say can produce your own fish and plants whole year round because you can schedule your stocking, you can schedule your harvest. Then number two is convert a small space in the premise of their house into a agri-aqua farm. Number three is a source of additional income for the family. And lastly is tourist attractors in urban communities. Since we are living a concrete jungle, it will be pleasant to the eyes if you can see some gardens and aquaponics unit in the middle of the cities. There, if you could see, there's aquaponic models of BFAR nationwide. So as of December 2020, BFAR already have 16 aquaponic models, ranging from backyard to commercial type aquaponics. Since we have many models for aquaponics, the price range for each aquaponics differ. So from as less as $52 up to the high of $2,000, we can do aquaponics in our backyards and in our farms. So you can choose the models you want or that is acceptable to your space or to your budget. So BFAR as part of the community, the Bureau accommodates requests from local government units and other agencies for technical assistance through webinars, lectures, collaborations, site evaluation, and to some extent, providing them aquaponic units. So positive feedbacks has been received from these beneficiaries once they realize the benefits of having an aquaponic setup in their backyard, from having readily available food for their own family, for their own home consumption, to being a source of extra income when excess produce is sold. And empowering Filipinos to produce their own food. The COVID restrictions to communities resulted in food shortage for households. With a small backyard or any space, coupled with the knowledge of setup and management and manage an aquaponic unit, people can have a readily available food throughout the year without even leaving their premises or houses. So in summary, with the pandemic that we're experiencing today, people now value food more than material things. Global and national economic growth has slowed down, and the cost of production and transportation of these food products will continue to be significantly higher than it is today. And for Filipinos affected by this pandemic, food resiliency is one way to combat the hardship brought by this situation. Producing our own food right in our own backyard through aquaponics can aid us in surviving this global adversity. So from the Philippines, this is our team from the Bureau of Fisheries and Aquatic Resources. So that's the end of my presentation. Thank you, and mabuhay from the Philippines. Thank you.
Okay, thank you, Dr. Joseph, for your nice presentation. Before moving to question and answer session, I would like to introduce Dr. Barbara Hostins, who is an chief scientist working for the NBA Aquaculture, and he is also a biofloc expert, having the hands-on experience in Brazil and also in other Latin American countries. So, Dr. Barbara, please say hello to our participants.
Hello, good morning, good afternoon for everyone.
Thank you. And before moving to a question answer session, I would like to request other panelists, if we appreciate if you could answer some of the questions through the Q&A tab, it will be much appreciated. And we also request the participants to respond our poll questions. Your response will be very much appreciated, and it will be helpful to evaluate our program. Thank you. So now we will go to the question answer session. First, I'd like to start with Dr. Barbara. Yes, as we see in Asia Pacific region, many aquaculture technologies are evolving like as Dr. Farshad also discussed one new technology today, mixotrophic aquaculture system. He's mainly focusing on the intensification of the farming system. So we also know about the biofloc, RAS, and other organic aquaculture systems. So what in your what do you think which kind of aquaculture technology will be cost effective and sustainable for our part, this part of this world, I mean Asia Pacific region?
So I think talking about the different systems, it also depends, like it was talked in the previous presentations, and it also depends on what the farmer will have available to invest, what is the reality, because it can vary, it's diverse, it can vary from farmer to farmer. I think the RAS costs are, we know that they are very high. Bioflocs is also initial costs, initial investments are very high, but it all depends as well on what the farmer has available or is willing to invest in this kind of systems and the returns they will have, of course. For shrimp culture, of course, if we compare it to salmon, it's much less. It's much less. How do you say? It's much less expensive to produce. So RAS system would increase the costs of maintenance and in initial investments, but it will all depend also in the technical knowledge available, because you cannot also invest a lot of money on a system and you don't have the knowledge to manage it, the right technical capabilities.
Okay, thank you. Thank you for the answer. So we'll go next question to Dr. Barbara. And we see biofloc technology is a new trend, new and emerging trend in Asia Pacific region. And also we see that this biofloc technology, that is the trend of biofloc technology, is increasing day by day. So do you think countries should adopt SOP for biofloc technology, and the all this, the countries which are practicing biofloc, so they should adopt some policies for the aquaculture water waste treatment facilities? You are muted.
Do you hear me well? Because I had some cut on the question, but
yeah,
okay.
So I have to repeat the question, or you can answer the question. Hello.
Hello?
Yes. Yes, please.
Hello?
Yes, we can hear you, Dr. Barbara.
Sorry, my connection just, my connection just broke. But okay, no, I heard the question. And I think first, talking about the wastewater management policies, I think it's something that should and probably will be implemented as a policy in the near future, considering the increasing.
Again, the line is breaking, Dr. Barbara. Okay, so we'll come back to Dr. Barbara later if you got time. I think we can move to other panelists because we are already running short of time.
Sorry for that, my connection just decided to break now. But do you hear me now?
Yes, please.
Yeah. Please, so, not talking about the waste management, this is a...
I think the line is breaking. We'll come back to you, Dr. Barbara. Please, we are moving to our first panelist. Well, already Dr. Odun left because he has another FAO webinar. So we will start with Dr. Martin. Yeah, Dr. Barbara, we'll come back to you.
I'm trying to fix it.
Yeah.
Yes. Okay.
Dr.
Do you hear me now?
Yeah, we'll come back to you. Yeah, later. We will come back to you later. So we will start with Dr. Martin. We received many questions for Dr. Martin already. I think the presentation was very interesting and very informative. So many questions from the participants. First, Dr. Martin, is genetically improved aquaculture commodity safe to eat? The first question.
Yes, absolutely. They are. All of these technologies that I was outlining, they are utilizing just naturally occurring genetic variants. So what these systems are doing is the same that has been done in livestock for many, many years already, for decades. And the point is that what we are doing is that we are just increasing the frequency of the favorable alleles or the favorable genes for the traits, and we are reducing the frequency of those that has a less positive effect. So we are not adding anything to the genetics, we are just utilizing the natural genetic variation. And this is accounting for today 99% of all the products that are developed through selective breeding. They are utilizing these type of technologies. So the fundamentals here, these are not GMO.
Okay.
These are not genetically modified organisms. So it's very good that these questions comes up so we can make that very clear.
Okay, very good answer. Thank you very much. And we,
but can I just add one thing? Because I also
Please.
picked up a very interesting question from Graham Mayer. Maybe I will take the opportunity to answer that now.
yes, please. Yes, please.
Graham is asking whether there are, what type of bottlenecks do we have on this uptake of these advanced technologies? And of course, I should make that very clear. What I was talking about is the kind of the front on the development on the technologies, and these most advanced genomic technologies, certainly they are going to be applied now in the most economically advanced production. So we are talking salmon, some of these high value species like shrimp, these technologies will come. But for many of the other species, of course, the majority of the aquaculture world is producing on species that doesn't have the resources to utilize these most advanced tools. So a very clear point I would like to make, and that is addressing this thing that Graham was referring to.
Okay.
Conventional breeding programs are extremely effective. So these genomic technologies is just some additional tools in the toolbox. But the fundamental changes, like the one I gave the example on the tilapia, and you see how this growth was improved over time. There was no genomic information used whatsoever. So the point is, we just use common sense. We design conventional breeding programs, and we have to ensure that we are setting up appropriate basic training in the fundamentals quantitative genetics, and having this technology transfer programs. I think that's a responsibility we have across the whole aquaculture society, that we are able now to share these fundamental knowledges, because some of these programs can be implemented at a fairly low cost with a very high return on those investments that are done. It all comes down to have the fundamental understanding of the genetics playing out in these systems. But this can be done without genomic information. So let me just make that very clear. I should have said that during my presentation too.
Okay, thank you. Thank you, Dr. Martin. You have another question, very good question. It is, I think, what is the difference between SPF? What is the difference in growth between SPF and SPR? Dr. Martin.
Well, first of all, let's just briefly define what we are talking about. SPF is specific pathogen-free materials. It says something about the health status of the animals. SPR, the products that Benchmark is developing, they are all SPF, but they are also combined with SPR, their specific pathogen resistance, because they are genetically enhanced their resistance to this key pathogen. So that's the key point. One challenge in Vanamai, in particular, is that there is an unfavorable genetic link between fast growth rate and the ability to withstand viruses. This is very evident in white spot. You can see that if you are selecting only for faster growing animals, you are making these animals more susceptible over time, and vice versa. If you select only for white spot, then of course there is a trade-off in terms of reduced growth rate. So the question really relates to this diversification also that is needed on the products, because if you have an environment which is low in the pathogen load, that's why you should then focus on the fastest growing lines. But we also have these intermediate lines which are combining the robustness with developing more tolerant lines. That's the SPR approach, and that is what is brought in through this approach that we are taking in Benchmark.
Okay, thank you very much. I'd like to ask you one additional question, moving to the next panelist. Would the gene editing be a more effective or faster way to produce a line of shrimp with a higher degree of disease resistance? Gene editing, yeah.
Yeah, it could be. I mean, gene editing is a relatively new technology, but it has a fantastic potential. Because through gene editing, what we can do is to go into the genome and identify those areas which are having these major effects in a positive way. And you may actually change very precisely some of these genes or the combination of alleles in those systems very precisely. So some of these changes that you can do through gene editing are not distinct. You can do the same type of changes with gene editing that you can do with selective breeding, but you can do it quicker. So that's a little bit on the backdrop there. So it is a very promising technology. However, there is a lot of issues, not issues, but there are some obstacles here. First of all, you have the legislation and the regulatory framework about these technologies, whether these are going to account as GMO products coming out, although they are not distinctive. In many cases, you cannot see the distinction in the product between a product developed by gene editing or by conventional selection. So that's a big question, and there's a lot of moving now in both in the United States and in Europe now on the legislation on these things. From a biological point of view, however, we also have to have a kind of target for making those changes. For example, if you have the IPN situation for salmon, I show that that's a trait that is determined by a single gene or a single allele. So the point is you have a major QTL affecting that precision and of course, or the expression of that resistance. In that case, we know the underlying fundamentals on the functional genomics, so you know where to target.
Yes.
But in most other cases, for example, in white spot, I show that this is a highly polygenic trait. So we do not fully understand where should we actually target the gene editing. So that's why when we are working on genomic selection, we are utilizing all these polygenic effects across the whole genome.
Yeah.
We are not targeting specifically one region because you don't find one region that is accounting for the differences expressed on this trait.
Thank you.
So there's a lot of functional genomics work that has to be done before you are preparing the grounds for doing gene editing. And we are doing in salmon, we are doing fundamental research on functional genomics. We have also initiated that in Vanamai, but it's a way to go before we have the very clear target for what type of gene editing modification that we need to do. We don't have the fundamental understanding of the functional genomics at this point.
Okay, thank you, Dr. Martin. Thank you for your time. And we appreciate if you could answer some of the questions through Q&A tab, or either we can send the question later to you also. You can respond by email to our participants.
Yeah, please do. Please send me those questions. I'm going to answer every single question for me.
Yeah, okay, thank you. So we are moving to our next panelist, Dr. Farshad Shishasian. Dr. Farshad, as you know, yes, mixotrophic aquaculture requires less oxygen, and it also need the fewer carbohydrate addition, and it also generates less oxygen and produce lower microbial biomass other than the other system like autotrophic or heterotrophic organism. So what are the parameters under which fish or shrimp farmer can encourage more growth of mixotrophic organism?
Okay, I don't think so. That is maybe true.
Specifically, what I'm asking about the parameters, specific parameters.
Yes. Okay. I mean, like, okay, when we talk about what is all about mixotrophic, it's going back to the original condition of these animals, that they are living in a natural condition. So it's a different type of consuming of the energy, and you have the whole things. You see, in the common RAS system, you are removing a lot of things from the system. So basically, you have water, but here we have phytoplankton, we have zooplankton, we have all these creatures together with the shrimp or fish. So here, you know, what we most consider is of course, the consumption of the oxygen is very, very important, because oxygen become one of the limiting factor, because you have a competitor for shrimp or fish to consume oxygen. And since we are doing a very high density, definitely oxidization has become a key things. And then when we talk about, like, you know, there is a stress management there. So it would be very different between the animal that they are like 100 pieces of shrimp in 1 square meter, or you have 2,000 pieces. Definitely there is a great amount of the stress in the system. How we manage this stress, I mean, like, I call it more like a stress management rather than the shrimp farming, because this stress can cause disease, can cause a lot of issue. Then you have a huge amount of the organic matter goes to a very small system. So then your ammonia, nitrite, nitrate, and also, you know, the bacterial load in the system, it's become very important. So the bacterial load can help us to prevent the disease in one hand, because if you develop the bacteria that are more beneficial to us, you have less, for example, if you talk about shrimp, you have less Vibrio. Like in my farm, I never face a Vibrio problem. And frankly speaking, we don't have, you know, ammonia or NO2 problem. Mostly we have a very big accumulation of NO3, so we reduce that in a different manner. So the factors that are important, I definitely, as I say, is a ORP, is a pH, of course, is a nitrogen, total nitrogen and the nitrogen content. And plus, you know, we look very closely to the mineral content, because minerals are extremely important. I know, am I able to answer the whole thing or not?
Okay, thank you for the answer. Next question for you from the participants is the cost benefit ratio of RAS, and what are the feeding strategy? What are the feeding strategy?
Very good question. I mean, like in terms of cost benefit, I would put it this way. You know, because I manage both type of farming. I have a RAS, I have an outdoor farm. So when I look at the cost, in a reality, if you can manage the proportion of the production, it is not really, I mean, like if you want to build a farm like a salmon farm, yes, you can never ever be able to, you know, get the correct ROI. That's what we modify it. That's what we make it a simpler and cheaper. That's what I can manage to make a profit out of my farm. If you are thinking that, I don't think so, nobody will produce RAS to sell like a 1,000 ton shrimp. So what we do with our shrimp, we sell a chilled shrimp every day to the market. And that is one of the key things. You cannot just do one in, one out, and you cannot compete with a conventional farmer that the cost of production is $1.50. In any case, my cost is a bit higher, but I am able to sell every day fresh animal. And then the next one, what was that you asked?
Sorry?
The cost benefit, and there was another thing.
Feeding strategy.
Feeding, okay. Feeding strategy. In my system, I do a lot of feeding. A number of feeding per day, it's a lot. It can be 10, 20, 30, depends on the size of the shrimp. And we don't throw the feed in the system. We have a specific location, like a long tray. So we pour the feed inside that, and then we let the animal to feed inside that. So then we have a better judgment about the, you know, the amount of the feed that give. I mean, like when you talk about the automatic feeder for RAS, it may work for shrimp, may work for fish, but when it's come to the shrimp, it is not that simple. So it's still, I prefer manual feeding, but in a controlled area.
Okay, thank you very much, Dr. Farshad. So I would like to I'd like to ask you one additional question.
Sure.
Are there any disadvantages in mixotrophic system?
I mean, it is really, I mean, like there must be no disadvantage. I mean, the disadvantage is that, let me I put it in a very simple way. When you do a mixotrophic, or you do bioflocs, or you do green system, or you do clean system, of course, each one of them has own, you know, the easy part and the complicated part. You see, I don't call disadvantage. We are not doing anything that is out of the norm. But the easy part in my system is you can manage water quality parameter is much more simpler and in a lower cost. A complicated part, of course, is a transaction and the difference between you have a phytoplankton, you have a bacteria in a very little area. And it works like sometimes like a bomb. Because if you don't manage it very well, the whole things can explode and your system can collapse.
Yes.
You see, that is the whole point. Of course, this kind of system, it's required years, you know, experience and understanding. Yes, it is not a book, you know, that you can read and do. You know, I always say, I am doing this 30 years. After so many years, you know, I mean, we say by joke, you have to be able to talk to the shrimp. And talking shrimp, you cannot find in any book. It means about the part of the art in aquaculture. Aquaculture is 70% science and 30% is art, which has not returned yet. So that's why sometimes we think things, we feel things that is true, but it's very difficult to explain. It's not that there is no explanation, but we don't know. But we learn by factors that these things can happen or cannot happen. That is the art part.
Okay, thank you, Dr. Farshad, and thank you
You're
for
welcome.
joining us. And we are now moving to our next panelist, Dr. Joseph. Dr. Joseph, you have some question from the participants. Why is that household families of Filipinos farmers are somewhat reluctant to adopt before aquaponics technology?
Okay, since aquaponics is newly introduced by the government, so some Filipinos want to see result first before they adopt. So that's the nature of people adopting new technologies. They want to see result first. So that's why the government is spearheading information drive and campaign to show the Filipinos that aquaponics is really a good alternative or an innovative way to produce food during this time of pandemic.
Okay, so next question, do you think any reason behind that, the reluctancy of the people for receiving this technology? Any specific reason?
Yeah, if we consider the situation right now, the reluctancy falls between because the materials for the construction of aquaponics should be bought outside of their home. So with this lockdowns and limited access to stores and malls, some Filipinos do not have access to materials. So maybe if the situation better on the future, maybe there will be more adapters for this technology.
I got it. So next question to you that from the participant, what are the challenges in aquaponics?
Challenges, yeah, that's a good question. If we for aquaponics, we will consider two biological factors. First, the plants and the fish. So the challenges is what combination of fish and plants best produces for your unit. So that's why we're doing some experiments to show the people what combination is good for this kind of plant or this kind of fish.
Yes. So an additional question with this, how do you manage fish health in aquaponics?
That's a good question. Since urban aquaponics is just in your backyard, we do recommend constant cleaning every day, just removing the weeds or the dirt on the units. That helps a lot. And of course, for sophisticated units, we recommend to monitor the water quality every 2 days or every day if you can.
Yeah, thank you.
Thank
and,
you.
I would like to ask you the last question.
Yeah, sure.
Do you foresee a time when aquaponics system will not only be installed as a way of food security, but as a mean of commercial activity in the rooftop garden or in the the barren public land? Do you how many times or how long it will take? What do you think?
Yeah, yes, we do expect it. With the government spearheading the information drive and technical assistance, many will be encouraged to invest in aquaponics, not only in the urban areas, but also in the provinces, especially if they know that the government will be there to support their activities from starting their units up to the marketing stage. So they'll be encouraged to adopt technology of aquaponics.
Thank you, Dr. Joseph,
Thank you.
For joining us. Again, I'd like to go back to Dr. Barbara.
Dr. Okay, Barbara? Now I keep my video off. Sorry for the. Okay, it was a sudden connection break.
Yeah, if you would be able to answer my last question was to you, that we see that in our region, the biofloc technology is very is booming day by day in every year, and it is very popular among the young entrepreneurs. So, do you think government should adopt any SOP for this, or and should adopt any policy for aquaculture wastewater management?
So starting from the waste management, I think it's, I believe that this will and should probably be implemented as a policy, because I think with the new technologies, new management techniques that we have increasing, and with the intensification of the cultures, like it was said before, we are solving the problems and managing the problems in the culture, but we need now to migrate and to go and solve the problems after, what we do with this waste, because also the shrimp toilets, this discharge of waste. So this needs to be implemented as an official policy. And I think there are increasing concerns about that, about the SOP for bioflux systems. I think, like it was said before by the other panelists, every country who have their own SOP, their own management, every country, every farm even, according to their own realities. The practices are according to their own realities. What I believe is there should be guidelines, of course, in each culture system, in each technique or management that you choose, and then talking about bioflux too. And also investing in technical knowledge for the people working in the farms more and more, that this becomes a common practice for shrimp culture and in general.
Okay, thank you, Dr. Barbara.
Thank you.
As we have running short of time, and we are already exceeded our limited time, so I would like to thank all the valued panelists present here today, and also I would like to thank our sponsor of this virtual event today, Benchmark Group, for their kind sponsoring this event. And in the conclusion, we can say that with all these presentations, question and answer session, and panel discussion, and also the comments from the participants, we try to address some of the recent innovations for the sustainable development of aquaculture in the Asia Pacific region. And we do believe that by establishing the governance on the sustainable development, and by applying the growth and resistant line of genetic selection, SPR and SPF shrimps or broodstocks, and intensification in the shrimp farming, of course, in a sustainable way, uh, we can uh, we can help the industry to make the to run in a sustainable way. So thus we can conclude here that these importance of technological developments and the technological development is necessary, though the climate change and local demand and the international market compliances is essential to assure the food security and the sustainable aquatic food production in the Asia Pacific region. So from this webinar, we can say that the technology and the sustainability will be the key in the future aquaculture development in the Asia Pacific region. And having said that, I would like to invite you all in our INFOFISH is organizing a couple of webinars under the Technological and Innovation series. Next week on the 27th of January, we are commencing innovations in seafood processing and packaging. Please keep following our INFOFISH, www.infofish.org website, and our social media sites for the recent updates in the seafood sector. So please stay safe and stay healthy, and till then, bye-bye. Thank you. Thank you. Thank you all.
Thank you. Bye-bye.